Good afternoon. My name is Peter Demuth from Elicio Therapeutics, and today I'll be sharing with you some of the preclinical program data for ELI-005 as part of our Science Advances publication, a lymph node targeted Amphiphile vaccine for inducing potent cellular and humoral immunity to SARS CoV-2. So initially, I'll start off with a bit of background on the lymphatic system. The lymph nodes are part of the lymphatic system, and this is an interconnected series of vessels and nodes throughout the body, which serve a primary function in detecting invading pathogens and coordinating and organizing protective immune responses. So you can think about the lymph nodes almost like the schoolhouse for the immune response. It's a site where numerous immune cells gather in large numbers and high densities, and it contains very specialized functionalities for detecting invading pathogens and coordinating the immune response to protect the host from those invading challenges. So with that in mind, how do we use the lymphatics and the lymph nodes to our benefit in the development of vaccines? And to get into that topic, we have to think about where vaccines go after they're injected. And it turns out that molecules that are injected into the peripheral tissues can be biodistributed to one of two sites. They can either be reabsorbed back into the blood and then circulated through the systemic circulation, largely to immunologically irrelevant sites. And importantly, molecules that take this pathway are not distributed through the lymphatics or into lymph nodes very efficiently. The alternative is absorption through the lymphatic vessels and concentration through the lymphatic flow into the lymph nodes. And what determines whether a molecule goes into one or the other of these two pathways to first order is molecular size. So things that are relatively small in molecular size are small enough to permeate the physical barriers that surround the blood vessels in the injection site. And this leads them to be taken up very efficiently back into the blood and again circulated through the systemic circulation. Things that are too large to be taken up back into the blood are instead accumulated into lymph vessels. And these materials eventually make their way and are concentrated into lymph nodes. And here they're efficiently able to interact with immune cells. And this is part of the natural mechanism the body uses to collect information from the peripheral tissue to organize and coordinate responses that will be protective. And you can see some of the evidence for this mechanism on the right portion of this slide. Unfortunately, many of the vaccine components that have been used traditionally are of relatively small size. So between 0 and 20 kilodaltons in molecular weight. And these fall into the category of molecules that, because of their size, are accumulated back into the blood from an injection site primarily. The opportunity exists, however, to reprogram the biodistribution of these molecules for more favorable uptake into lymphatics if we can increase their effective molecular weight. And on this slide, what you're seeing is endogenous tissue albumin. This is a very ubiquitous protein present in the tissues, which, because of its larger molecular weight, is almost universally accumulated from those tissue sites into the lymphatics. And so the idea behind this program and the technology here at Elicio is to reprogram the biodistribution of these conventional vaccine components through association with albumin to enable them to more efficiently traffic from an injection site into the lymphatics and thus engage the immune response much more potently. So that's what we'll be talking about today through the course of the review of the data on this program. So let's talk a bit about the ELI-005 product candidate itself. This is made up of two components, one being the antigen. This is the receptor binding domain from the SARS CoV-2 spike protein. It's an antigen that's present on the surface of the viral capsid and is responsible for mediating the interaction with host cells in the initial stages of viral entry and infection. It's also known to be the target of neutralizing antibody responses, which can be protective. and a target for both CD4 and CD8 T cells, which again can contribute to either prevention of disease or a mediation of the severity of disease. And importantly for our purposes, because this protein is approximately 34 kilodaltons in size, it's predicted to have a suitable lymph node targeting profile to accumulate into lymph nodes and be a suitable antigen for a subunit vaccine strategy. The second component of the vaccine is a lymph node targeted form of the adjuvant CpG. This is a potent TLR9 agonist, which without any modification in its conventional form is of a low molecular weight, which would predict not accumulation into lymph, but instead recirculation through the blood. And the key modification that we've made in our candidate is to include this lipid tail, which we use to improve association of this CpG molecule with endogenous albumin in the tissue at the injection site, which, as I mentioned, has this very efficient ability to enter the lymphatic vessels and then accumulate into the lymph nodes. So the idea here is to improve the lymphatic targeting of the adjuvant itself in order to improve the overall activation of the immune response and orientation of it towards this target in a vaccine setting. So the mechanism is shown here on this slide. These two components are injected into a peripheral tissue site. The adjuvant at this site quickly interacts with albumin, which is present within the tissue at high levels. And this effectively increases the molecular weight of the adjuvant, and albumin is then able to chaperone it through the lymphatic vessels directly into the lymph nodes. In parallel with this, the antigen itself, because of its larger molecular weight, is also likely efficiently trafficked through the lymphatic vessels into the lymph nodes. And these two components together within the lymph nodes offer the opportunity for very efficient uptake into antigen-presenting cells and potent activation of those cells, as well as efficient communication of activating signals to T and B cells, which ultimately will provide the protective functional response against a potential infection. So let's walk through some of the data. In this slide, you can see a variety of different groups which were compared in a series of preclinical studies in mouse models. Just to orient you to that, on the left here, we have animals which were treated with mock vaccination, animals which received vaccine formulated in alum as a benchmarking group. Alum is a very well-known and well-studied adjuvant used in the clinical space and represents a really important point of comparison for subunit protein vaccine candidates. In addition, we tested soluble CpG, as well as our Amphiphile CpG or AMP-CpG, which is the component used in the ELI-005 product. The important inclusion of both of these allows us to make comparisons in the immune response, which are derived from the improved lymph node targeting of the Amphiphile CpG relative to the soluble CpG. And the data here initially is showing you the strength of the CD8 T cell response that we can induce targeting the RBD antigen. And you can see here on the left the enumeration of cytokine secreting CD8 T cells, and then the example flow plots here in the middle, and then a summary of those results here on the right. And as you can see, where alum is producing barely responses above background, soluble CpG is able to mediate the induction of responses which are detectable, around 10 to 20% of all CD8 T cells producing either TNF or interferon gamma, while the Amphiphile version of this vaccine, the ELI-005 candidate, is producing substantially higher numbers, around 40 to 50% of all circulating CD8 T cells in the blood are specific for epitopes within the RBD antigen. We've also looked at responses to the antigen induced in the CD4 T cell compartment and see that the trends are largely the same, with little to no detectable CD4 T cells produced in the alum vaccinated animals, while soluble CpG vaccinated animals produce some measurable CD4 T cell response, which is enhanced in the setting of AMP-CpG vaccination, as you can see here. In addition to looking at the peripheral blood, We were extremely interested in this series of studies to look at the primary tissue of viral exposure, the lungs, to determine whether responses at this local tissue site could be induced. And the motivation here is to produce a population of T cells in a site of first entry that could be rapidly able to respond to a nascent infection. And similar to what we've seen in the blood, in the lungs, what we observe is that while alum produces little to no CD8 T cell response specific for the vaccine antigen in the lungs, soluble CpG produces a measurable T cell response, which is further enhanced through the inclusion of a lymph node targeted version of that CpG in the Amphiphile treated animals. And here, impressively, these responses are rising to between 70 and 80% of the total CD8 T cells present in the lung compartment. And similar trends are observed if we look at CD4 T cell responses. Again, alum producing little response that we can detect. Here, interestingly, in the soluble CpG case, there's no statistical enhancement of CD4 T cells targeting RBD in the lungs, while AMP-CpG vaccination in the ELI-005 candidate is the sole vaccine treatment regimen that was able to produce detectable levels of RBD-specific CD4 T cells. In addition to looking at TNF-alpha and interferon gamma through ICS assays, we conducted a series of studies and restimulation of cells collected from lungs to look at various other cytokines produced in response. And what we observe in these studies is, again, a significant production of interferon gamma in the Amphiphile CpG vaccinated treatment group. This is also seen in TNF-alpha, two important Th1 cytokines, while in the setting of IL-6, IL-4, IL-10, or IL-17, we don't observe any production of these classic Th2 or Th17 cytokines, indicating that the cellular response induced by the vaccine is Th1 biased. In addition to looking at cytokine production, we were interested to look at the T cells that were secreted into the lung fluid, here looking at bronchoalveolar lavage. In both the CD8 and CD4 settings, we were able to observe that all vaccine candidates increased the number of T cells for CD4s, while only the Amphiphile vaccine in this setting was able to produce CD8 T cell enhancement. And the trends observed here indicate with the vaccines a transition of the phenotype of these cells from a naive to an antigen experience and effector memory T cell phenotype, both in the CD8 and CD4 compartments, suggesting that these cells are not only present in the lung tissue, but may also be present within the fluid of the lungs, providing a further level of protection to this site of first viral exposure. Let's shift our attention to antibody responses, the second arm of the immune response, potentially important for providing protection in this setting. You can see in assays of neutralization activity that alum, soluble CpG, and AMP-CpG produce high levels of neutralizing activity, about 100-fold or more higher than what's observed in convalescing human patient samples. We can see that this is consistent when we look at serum IgG levels, with the one interesting differentiating factor in these responses being the Th1 versus Th2 bias. And just to go into that a bit more detail, here what you can see is while alum is producing mostly Th2 bias in the IgG response, and soluble CpG is producing more of a balanced Th1, Th2 profile, the AMP-CpG vaccine treatment group is exhibiting an extremely strong bias for Th1 isotypes of IgG, favoring the 2C isotype versus the 1 isotype, suggesting that AMP-CpG provides a more Th1 biased response in the IgG compartment. So the next series of studies were intended to look at the opportunity to reduce the amount of vaccine and evaluate whether reduced doses of vaccine could induce a similar level of T cell and antibody response. The motivation, of course, being the opportunity to vaccinate more patients with the same amount of gross material. And so here again, we've compared alum and soluble CpG vaccines given at 10 micrograms antigen dose to AMP-CpG adjuvanted vaccines with the RBD antigen administered at either 10, 5, or 1 micrograms dose level. And in this readout, we're looking at the level of T cells within the spleen, which are specific for the RBD antigen. And interestingly, we observe again this hierarchy in the 10 microgram dose level, where alum and soluble CpG are outperformed by the AMP-CpG vaccine. And interestingly, we see very little drop off in the strength or magnitude of the T cell response detected in the spleen as we move from 10 to 5 to 1 micrograms antigen dose. In addition, when we look in the blood for CD8 and CD4 T cells responding to the RBD antigen, we see a very similar trend. Again, this hierarchy where AMP-CpG is driving much higher levels of cytokine secreting CD8 and CD4 T cells, and very little, if any, statistical drop-off in the magnitude of those responses in the 10-fold dose de-escalation from 10 to 5 to 1 micrograms, suggesting that it is possible to maintain the strength of T cell response with a much lower dose of antigen when it is combined with AMP-CpG adjuvant. This phenomenon was also observed in the lung readouts. So here you're looking at CD8 and CD4 T cells in the lungs. You see this similar hierarchy of response at the 10 microgram level. And again, very little, if any, statistical drop-off in the magnitude of these responses as you de-escalate the dose of antigen, both in the CD8 and CD4 T cell compartments. So this suggests an opportunity to maintain the strength of the T cell compartment, both in the blood, the lung, and the spleen with greatly reduced amounts of RBD antigen if it is combined with AMP-CpG. In addition to looking at T cells, we also looked at neutralizing activity in the antibody compartment. And here again, consistent with our prior data, we see no statistical difference between the alum, soluble CpG, and AMP-CpG vaccine candidates. And interestingly, as we dose de-escalate of the RBD in the AMP-CpG vaccine candidates, again, we see no statistical decline in the magnitude of that response. And it's maintained at levels much higher than we observe in convalescing human patient samples. Again, I'll just highlight this differentiating aspect of the IgG response, where we do see that AMP-CpG is able to drive greatly enhanced Th1 bias in the IgG compartment relative to that observed in the alum and the soluble CpG responses. So beyond looking at responses in young healthy animals, we were motivated to look at responses in elderly animals as a model for the potential response we could induce in elderly human patients, which are challenging because of immune senescence or the age-related decline of their baseline immune function, and also disproportionately suffer more severe outcomes in COVID-19. So for these studies, we looked in aged animals, again comparing soluble CpG and alum to AMP-CpG vaccination, which is given, again, either at 10, 5, or 1 microgram RBD antigen dose level. So here we're looking at CD8 T cell response levels in the lung and observe that, again, alum and soluble CpG produce relatively low amounts of responsive T cells within the lung compartment, while AMP-CpG is maintaining the level of response that we observed in young animals between 60 and 70% of CD8 T cells responsive to RBD. And this is not statistically significant in decline as we reduce the antigen dose level from 10 to 5 to 1, suggesting again the potential to dose de-escalate the vaccine in the AMP-CpG setting without any sacrifice in the strength of the CD8 T cell responses. This trend is maintained if we look at CD4 responses, again in the lungs with very high levels of CD4s in the AMP-CpG group, and again, no statistical drop-off as we decrease the level of the RBD antigen. Switching our attention back to the neutralization and Th1 versus Th2 bias of the antibody response. In this setting of aged animals, we see that AMP-CpG at the 10 microgram level is able to drive significantly higher levels of neutralization activity compared to alum and soluble CpG. And this is maintained even at lower antigen levels of 5 and 1 microgram, again, greatly in excess of what we observe in convalescing human patient samples. And the similar trend of Th1 bias in the AMP-CpG treatment groups compared to what we observe in alum and soluble CpG. Finally, we were interested to look at the durability of these responses. And to do that, we monitored peripheral blood T-cell responses for the course of several weeks following the completion of the vaccination regimen, again comparing alum, soluble CpG, and AMP-CpG. And consistent with our prior results, we observed again this hierarchy of response magnitude, with AMP-CpG producing higher dose or higher levels of T-cell response. And interestingly, we observed that these responses were maintained for the period of study included in this experiment. In addition, we looked at endpoint analyses for spleen and lung T cells on day 70, several weeks after completion of the vaccine regimen, and observed at this extended time point elevated levels of cytokine-producing T cells, both in the spleen and the lung, suggesting that these responses can be maintained through a vaccine regimen for a period of several weeks following completion of the regimen. Similar to the T cell responses in the antibody compartment, we observed a similar level of maintenance of response with no statistical decline across all of the comparator groups between day 49 and day 70, suggesting that these responses can be maintained at least for the period of study included in this particular experiment. So to summarize what you've seen in the presentation, we've evaluated the ELI-005 candidate in a number of studies looking at the cellular immune compartment. We've seen very impressive levels of CD8 and CD4 T cell response. These are polyfunctional Th1 cytokine secreting T cells. The responses that we observed were maintained, even with a tenfold reduction in the dose of spike RBD antigen used. And these responses were observed again, both in young, healthy animals as well as aged animals as a model for immunosenescence. And importantly, these responses were observed to be durable for at least 5 weeks following completion of the regimen. In addition to blood, we also saw substantial T cell responses within the lung compartment, with CD8 and CD4 T cell responses, both within the lung tissue and within the lung fluid. These T cells, again, were polyfunctional and Th1 in bias, and we observed a similar opportunity to reduce the spike RBD dose while maintaining the strength of those responses, both in young and aged animals, with observed durability for at least 5 weeks after completion of the vaccine regimen. In the antibody compartment, we observed high titer responses, which greatly exceed those found in human convalescing patients, with an interesting bias towards the Th1 isotype profile, especially in comparison to those observed in the vaccine comparator groups of alum and soluble CpG. And importantly, again, this opportunity to reduce the amount of spike RBD antigen while maintaining the strength and character of the response, both in young and in aged animals. Thank you very much.